Okay, we're in for two more treats this afternoon. Please take your seat. It is the privilege of the Science Co-Chair to introduce the Science O'Donnell awardees. And the first awardee is Dr. Vincent Tagliabracci from the UT Southwestern Medical Center, where he is an associate professor. Please, Vinny.
Okay. Well, first, thanks, Gaudens, for the introduction, and I'd like to start by thanking Dr. Lee, TAMEST, the O'Donnell Foundation, and Dr. Olson for being my nominator for this award. I'm deeply humbled and indeed honored to be named the 2024 O'Donnell Awardee in Biological Sciences. So my laboratory has had a long interest in a large superfamily of enzymes known as the protein kinases. These enzymes perform a relatively simple reaction. They bind cellular ATP, and they take the gamma phosphate, or the terminal phosphate, from the cellular energy source, and they stick it onto protein substrates, usually serine, threonine, and tyrosine residues on these proteins. Now, in humans, there are more than 550 of these enzymes that have been assembled into this beautiful evolutionary tree known as the human kinome. And the reason why these are important is because several of these protein kinases are actually mutated in different human diseases. Most notably, several of these are mutated in human cancers. And in fact, there's a very famous drug called Gleevec that was discovered about 20 years ago that was termed a magic bullet by Time magazine, where this is a drug that targets a protein kinase that's part of a fusion protein. It's called the BCR-ABL fusion protein, which is mutated in patients with chronic myelogenous leukemia. And the beautiful thing about this drug is that you just take one simple pill, and these patients went from a 5-year survival rate of about 5% to more than 80 to 90% survival rate. So this is essentially a cure for cancer. It was just one pill that was targeting one of these mutated versions of protein kinase. Now, in addition to the 500 or so active kinases that are assembled on this tree, about 10% of them, or about 50 or so, are considered catalytically inactive enzymes. And what that means is that they're missing key active site residues that are thought to be essential for catalysis. So these, because of the fact that they're considered catalytically inactive, they've been termed zombie enzymes. And I don't know if I'm 100% on board with that, but that's what people have called them. So over the past 8 years or so, my laboratory has taken a bioinformatics approach. And more recently, we've used artificial intelligence, such as AlphaFold, structural modeling. And we've essentially expanded the human kinome. We've found new branches on the human kinome tree that have really kind of redefined what a protein kinase can do. And as I told you, kinases are thought to transfer the gamma phosphate, or a single phosphate molecule, onto proteins. What we're starting to find is that these so-called pseudokinases, or inactive kinases, are actually active, but they're just performing a different chemistry. So one such example is a protein kinase called CIDJ from Legionella pneumophila. This is a bacterial pathogen that causes a sometimes deadly form of pneumonia in humans called Legionnaire's disease. We found that despite the fact that CIDJ adopts a three-dimensional protein kinase fold, it actually still binds ATP, but it uses the energy from ATP to transfer glutamate residues, so the amino acid glutamate, onto protein substrates. And this is a post-translational modification called glutamylation. Now, in addition to CIDJ, we've also worked on another predicted pseudokinase called cello. And this forms the basis of what I'd like to tell you guys about today. So cello was predicted in 2012 to be catalytically inactive, and the reason why it has the catalytic lysine residue that you find in canonical kinases. There's also a metal-binding aspartic acid residue that you also find in your normal protein kinase. However, cello was coined a pseudokinase because it's missing this catalytic base. This is the aspartic acid residue within this HRD motif. You could see in the cello proteins, there was a conserved HGV motif. So for many years, we worked on this. This was really just kind of a curiosity-driven project. This protein kinase is conserved all the way back in bacteria. You can find versions of this in yeast. And there weren't really any papers published on this protein. So at the time, Anju Srilatha, who was a postdoc in my lab at the time and is now here as a protege, she determined the three-dimensional structure of the cello protein, and kind of as expected, it did have this three-dimensional fold that looked like a protein kinase. However, what Anju noticed was that the nucleotide, the ATP within the active site, was essentially flipped by 180 degrees. Okay, so instead of it binding this way, it was binding 180 degrees the other way. And from these structural analyses, Anju was able to discover that the cello proteins aren't actually inactive after all. In fact, they're active, but instead of transferring this gamma phosphate from ATP, because the orientation of the nucleotide was flipped, it's now transferring the AMP group to protein substrates. And this was actually a post-translational modification discovered by Earl Statman back in the 60s when Mike Brown was actually a postdoc in his lab. And it was rediscovered in the early 2000s by Kim Worth and termed ampylation. So this was exciting. This was really the first paper that came out of my lab. We were excited. And then one sunny day in June, I woke up to this email. So this is June of 2019. And it's an email from a bioinformatician from France named David Cartlin. And he said, "Dear Vincent, I really enjoyed your article on cello. I found it by chance because I discovered that the N-terminus of the viral protein NSP12, called the Niran domain, is most probably homologous to it in a large superfamily of viruses, the nidoviruses, containing the deadly SARS virus itself in the family coronavirus." So some of you may remember back in the early 2000s, there was an outbreak of a version of COVID. It was SARS coronavirus 1. And mind you, this is in 2019, and he continues, he says, I'm an expert in remote homology detection and pretty sure that the Niran domain is homologous to the cello kinases and probably has a similar function and mechanism. And in the interest of full disclosure, I wrote back to him and I said, "Dear David, thank you for your email. However, we have no interest in studying coronaviruses." And then this happened. So everybody knows what happened in late 2019, early 2020. SARS-CoV-2, which is a causative agent of the coronavirus pandemic, has basically changed the way we live and has killed more than 7 million people. So we decided at the beginning we better start taking a look at this. We knew what the cello protein was doing. We knew the chemistry of the cello reaction. We thought that maybe we can try to understand what this enigmatic domain in the coronavirus protein was doing. So just a bit of background. The coronavirus is a positive sense single-stranded RNA virus, so it could enter the cell and it can be directly read by the ribosomes, translate viral proteins, which then facilitate the replication, transcription, and evasion of the immune system of the virus. So some of you might be familiar with the spike protein. This is what, if any of you have had the vaccine, this is the protein that's made within our body that elicits an immune response and confers protecting antibodies against future infections. There's also these non-structural proteins that are on the 5' proximal 2/3 of the genome, including this protein called NSP12. This NSP12 protein contains the RNA-dependent RNA polymerase. So this is the target of remdesivir. This is the drug that was frequently used in the early days of the pandemic that showed some efficacy. But attached to the polymerase is this really kind of unknown protein that people didn't pay much attention to called the Niran domain. And this is the domain that was similar to cello that David had pointed out to me in this email. So we thought, okay, this is kind of cool. I wonder what this thing is doing. Well, in the original SARS coronavirus that caused the disease in the early 2000s, it was known that if you made mutations within some of those key residues that I showed you earlier on, that the virus couldn't replicate. So that says that this domain is absolutely required for the function of the coronavirus. And importantly, when the NSP12 protein from SARS-CoV-2 its structure was resolved, the nucleotide, very similar to Anju's structure, showed this flipped orientation of the nucleotide. So to us, that meant that the chemistry of the reaction is going to be very similar between the coronavirus protein and the cello protein. Now, there was one other thing that I need to tell you, is that the RNA genome of RNA viruses has a 5' cap structure. So this is essentially a chemical camouflage that hides the RNA, the viral RNA, from the host immune system. It protects it from degradation by exonucleases, and importantly, it facilitates translation of viral proteins. So without this cap structure, the virus can't replicate. Now, obviously, this isn't a cap. It's a very specific chemical moiety, which is a guanosine residue linked by way of a 5' triphosphate residue linkage to the first nucleotide within the RNA. Now, in our bodies, there's what's called the canonical RNA capping mechanism. So when our DNA is converted into RNA, the RNA needs to form this cap structure, or else our cells will actually think that it's a viral RNA. And how this happens is the nascent 5' triphosphorylated RNA comes out of the active site of the polymerase, and it's immediately acted upon by an RNA triphosphatase that trims the terminal phosphate on the nascent 5' strand. This creates a diphosphate. The second step, a guanylyl transferase, could transfer the GMP from GTP onto the 5' end to create this, what's called core cap structure. So this is the guanosine with 3 phosphates linked up to the first nucleotide within the genome. And then in a subsequent step, there's an N7 methyltransferase that adds a methyl group to the N7 position on the guanosine. And then there's a 2O methyltransferase that methylates the 2-hydroxyl on the first base within the genome. Now, within the coronavirus genome, they encode the NSP13 protein, which at least in vitro has RNA triphosphatase activity. The NSP14 protein is the N7 methyltransferase, and the NSP16 protein is the 2O methyltransferase. So it was assumed that the coronavirus capping mechanism proceeds in a very similar fashion to the canonical capping mechanism, with the NSP12 Niran domain functioning as the guanylyl transferase. However, there's really no evidence to support this. So we had read these papers, and when the lockdown ended, we went back to the lab and we thought, well, what a great hypothesis. We should test this and see if we can get this to work. To make a long story short, this reaction doesn't happen. Okay? So then in early 2021, there was a paper by Slanina et al. Showed that the Niran domain could actually ampylate this other protein within the viral genome called NSP9. And this happens on the extreme N-terminus of the protein and forms what's called a phosphoramidate bond, so a phosphate group attached to a nitrogen. And at the time when we read this, Gina Park, an MD Ph.D. student in my lab, reasoned that because this domain is always attached to the polymerase, the concentration of RNA is going to be extremely high within the vicinity of the Niran domain. And she said, ATP also kind of looks like the first nucleotide within the genome, which is also an A. It has this 5' triphosphate, and you can see the ATP looks somewhat similar to the 5' region of the RNA. So Gina said, well, what if this domain is actually transferring the RNA chain onto a protein and not necessarily just an AMP group from ATP? And this would be a post-translational modification called RNAylation. So we said, okay, let's give this a try. So we synthesized a 5' triphosphorylated 10-mer corresponding to the first 10 bases of the SARS-CoV-2 genome. And Gina did this experiment, and this is one of those results that you dream of as a scientist. You come in and your student shows you this data. So here's the NSP9 protein, and when Gina adds NSP12 and just monitors by SDS page, so this separates proteins based on molecular weight, you can see as the time increases, so does the molecular weight of the NSP9 protein. And then we can add an enzyme that degrades RNA. This is an RNase. And when she added the RNase, you could see this band now collapse down to this band. And this reaction was entirely dependent on a functional Niran domain. And when we mutated the polymerase domain, nothing happened. So that basically what these results were saying is that this domain can take the RNA that's coming out of the polymerase and it can attach it onto another protein. So we thought this is cool, but what does this mean in terms of the biology of the coronavirus? Well, to get at that, we started reading on how other viruses, including the rabies virus, this is also the Ebola virus, they're all part of this order of viruses called the mononegavirales. And how they cap their genome is, it's very complicated, but they have this protein called the L protein that contains a domain called the polyribonucleotidyltransferase domain. Try to say that 3 times fast. And what it does is, as the RNA comes out of the polymerase, it attaches it to a histidine residue within the active site, and it also forms one of these PN bonds, or these phosphoramidate bonds. And this acts as an intermediate step for the next reaction to happen, which is to transfer the RNA onto GDP to form this core cap structure that camouflages the Ebola RNA, just like the coronavirus needs to be camouflaged. So we reason this is kind of similar to what we're seeing, but instead of the RNA being attached to the enzyme itself, the RNA is actually sticking the protein is actually sticking the RNA on another protein. So we said maybe if we just add GDP, maybe we can form this core cap structure. Well, so here's the experiment. So here's the NSP9 protein again that's separated on SDS page, and here's the NSP9 protein that's linked up to the RNA. Now, when Gina added just buffer, you could see, as expected, nothing happens. But when she spiked GDP into the experiment, you could see now, in a Niran-dependent manner, we can remove the RNA from the NSP9 protein and regenerate unmodified NSP9. Okay, so what's happening to the RNA? It's really the RNA is what we want to know. Does this RNA have this core cap structure? So this is a different experiment now. This is something called urea page that we can use to separate RNAs based on molecular weight. So you can see here's the 5' triphosphorylated RNA. And then when it's attached to the NSP9, it doesn't enter into the gel because it's too big. The protein can't enter into these urea page gels. But now when she uses alpha-labeled P32 GDP, you could see we can release the RNA into the gel. It's dependent on the Niran domain, independent of the polymerase domain, and this is a positive control. Now, because we're using a radioactive molecule with the phosphate group on the GDP labeled, we could detect this now by autoradiography. And you could see that when we expose this film, the only 3 lanes that have this P32 signal are consistent with this kinase-like, this cello kinase-like domain, adding the core cap structure to the viral genome. And just to go one step further, we can cut these bands out, and we could send them for something called mass spectrometry. And we can use a technique called liquid chromatography to separate the reaction products and identify by mass spec the core cap structure that forms only when we use a wild-type version of this domain and not a mutant version. Okay, so now how important is this for SARS-CoV-2? We teamed up with John Scoggins and his research technician, Jennifer Eitzen at UT Southwestern, who during the pandemic set up a BSL-3 safety facility in order to grow the virus and do experiments. And I think you guys can easily tell here, the wild-type virus replicates very, very nicely. But when we make any mutations within the virus, within any of the amino acid residues that are involved in forming this core cap structure, you can see the virus doesn't replicate at all. So what this means is that targeting this domain, and remember I told you at the beginning, a lot of drugs on the market are targeting kinases. So we feel that this is a kinase domain, and targeting it with a first-in-class antiviral inhibitor to treat not only COVID-19, but perhaps another version of coronavirus that may and likely will emerge in the future is a valuable option. Okay, so here's kind of the last slide on the mechanism on how this works. So the NSP9 protein can bind within the active site of this Niran domain. And this is the amino terminus of asparagine 1. This will attack the alpha phosphate of the 5' triphosphorylated RNA as it enters into the active site, release the diphosphate, form this NSP9 RNA intermediate that was migrating on the gel larger than the unmodified NSP9. Now, in a second step, GDP has to bind very specifically. I didn't show you this, but GDP is the only nucleotide that could be accommodated for this step. ADP, CDP, and UDP, which are chemically similar, don't work. So there's a very specific pocket that accommodates GDP, and now it's this phosphate group that can attack here, cap the RNA, and release NSP9 for another round of catalysis. Okay, so where are we going from here? So as I mentioned, I think developing a drug, an antiviral targeting this domain, I think would be a great idea. And to this end, we've actually developed a high-throughput screening assay to screen hundreds of thousands of small molecules in pursuit of an inhibitor that can bind to the Niran domain and inhibit its activity. So we've done that, and we've actually identified an inhibitor. And here's a structure of the Niran domain showing some density in the active site for a covalent inhibitor that we've recently identified that inhibits the Niran domain in vitro. And so now we're trying to work with medicinal chemists to optimize this compound with the hopes of one day developing a drug to treat coronaviruses. And with that, my last slide is, this is last Christmas. We went curling. If you've never gone curling before, it's a great thing to do. So Gina Park and Adam Ozinski were really the key drivers of the project on SARS-CoV-2 that I just showed you. And Genaro Hernandez in my lab, with help from Bruce Posner, have been involved in developing inhibitors. And then John Scoggins, and as well as several other my UT Southwestern colleagues. And you guys for sticking around and listening to me. So thank you very much.
Thank you very much. Do we have questions? Maybe while we wait for the mic, this RNA-ylation, right? Is that something that is worth now looking for also in mammalian cells?
Yeah, absolutely. We haven't been able to detect it. I know there's a recent paper showing that there's another virus that can kind of do a similar reaction, but on a protein substrate, has nothing to do with the capping. So I think that might be a little bit more common than previously appreciated, for sure.
Okay, cool.
I was trying to follow all the orientations there, but on silo, I understand why you want to transfer the AMP and why you have the different orientation on the kinase bind. But why do you need on Niran base? You're binding from the other side, right? So why it's inverted?
Yeah, so there's two binding pockets within the same active site. One binds in the flipped orientation with the phosphate groups in. So there's a basic patch that we've got structures of this now that accommodates the RNA, the triphosphorylated RNA. And then there's another more hydrophobic patch that accommodates the G for the GDP.
So you're telling me that although they have similar structure, silo and Niran will act completely different?
The viruses.
Okay, that's what I was trying to follow. Yeah. Thank you very much for clarifying. Thank you.
Eric.
How many more pseudokinases are out there? And it seems to me you're solving about one per year. So what's the future look like?
Thank you for that question. We've got a few more up our sleeve. Nothing that's as crazy. This is the craziest one that I've ever seen, what it can do. We've got a few more that we're working on. One is, two of them are actually lipid, turning out to be kinases that phosphorylate lipids. So we're kind of excited about following up some of those. I don't know if we could keep it up one per year, but I'll try my best.
Great. So if there is no more questions, let's congratulate Vinny one more time.